Prokaryotic DNA replication is a rapid and efficient process that allows bacteria to divide quickly. It occurs in three main stages: initiation, elongation, and termination. Here's a breakdown of the steps. Initiation, origin, recognition. Replication begins at a specific site on the circular bacterial chromosome called the origin of replication, or ORIC. This region contains specific DNA sequences that are recognized by initiator proteins. Initiator protein binding. Initiator proteins such as DNA and E. coli bind to the ORIC region. This binding causes the DNA to wrap around the DNA proteins, leading to localized unwinding of the DNA double helix. Helicase loading. The unwinding of DNA facilitates the loading of DNA helicase, known as DNAB in E. coli, onto the single-stranded DNA. Helicase is an enzyme that further unwinds the DNA double helix at the origin, breaking the hydrogen bonds between the complementary bases. Formation of replication forks. As the helicase unwinds the DNA, it creates two replication forks that move in opposite directions away from the origin. Each replication fork is a Y-shaped structure where the DNA is being unwound and new strands are being synthesized. Single-stranded binding proteins, or SSBs. As the DNA strands separate, single-stranded binding proteins bind to the single strands to prevent them from re-annealing or coming back together, and to stabilize the unwound DNA. Primase binding, an enzyme called primase, referred to as DNAG in E. coli, binds to the single-stranded DNA at the replication fork. Primase synthesizes a short RNA primer that is complementary to the template DNA strand. This primer provides a free 3' hydroxyl group, which is essential for DNA. The primary enzyme responsible for synthesizing new DNA strands is DNA polymerase III. It adds deoxyribonucleotides to the 3' hydroxyl end of the RNA primer, extending the new DNA strand in the 5' to 3' direction. On one template strand, the 3' to 5' strand, DNA polymerase III can synthesize a new complementary strand continuously in the 5' to 3' direction as the replication fork progresses. This newly synthesized strand is called the leading strand. Only one RNA primer is needed for the leading strand. On the other template strand, the 5' to 3' strand, DNA polymerase III cannot synthesize DNA continuously because it must move in the 5' to 3' direction. Instead, this strand is synthesized discontinuously in short fragments called Okazaki fragments. Each Okazaki fragment requires its own RNA primer synthesized by primase. DNA polymerase III extends these fragments. DNA polymerase I action. After the Okazaki fragments are synthesized, DNA polymerase I removes the RNA primers using its 5' to 3' exonuclease activity and replaces them with DNA nucleotides. It also has a 3' to 5' exonuclease activity for proofreading. DNA ligase action. The Okazaki fragments are initially separated by small gaps. The enzyme DNA ligase catalyzes the formation of phosphodiester bonds to join these fragments together, creating a continuous lagging strand. Proofreading. During elongation, DNA polymerases, primarily DNA polymerase III, have a proofreading activity. They can detect and remove incorrectly incorporated nucleotides and replace them with the correct ones, ensuring high fidelity of replication. Replication proceeds bidirectionally from the origin until the two replication forks meet at a specific region on the chromosome called the termination region. This region contains specific DNA sequences called TER sites, for example, TER-A through TER-J in E. coli. In E. coli, the TUS protein, terminus utilization substance, binds to these TER sites. The TUS-TER complex acts as a contrahelicase, meaning it can stall or block the progression of the replication forks. The TER sites are arranged in a way that allows the forks to enter the termination region, but prevents them from proceeding further in the opposite direction, ensuring they meet within this defined region. When the two replication forks meet, DNA replication is completed. The two newly synthesized circular DNA molecules are often interlinked, forming catenanes. Enzymes called topoisomerase I4 in E. coli separate the two daughter DNA molecules by transiently breaking and rejoining the DNA strands, resulting in two independent circular chromosomes that can be segregated into the daughter cells during cell division.